didn't know quite what to entitle the talk, because it really, I'm talking about the way scientists approach these kinds of issues. And EMF and DNA is what I ended up talking about. I could have said EMF and science. I could have said EMF and biology, which is a little more focused. But it really, I'll bring it down to the level of EMF and DNA. It's that part of biology which I think is really essential to us as living beings and as a species, as human beings. The next slide. I start off with a quote. It's actually, I'm quoting, but it's something a professor mentioned in graduate school, one of his courses, and I put it down. It's it's not there. Mother Nature guards her secrets well, but she never lies. And actually, this is something that Albert Einstein said in a talk at Princeton back in 1921. But he said it in German, and the translation is not exact. But I think you get the idea that Einstein was really summarizing the way scientists look at the world. They say you can do science, and when you do it, it'll be hard to extract the information sometimes. In fact, often it's very hard to extract the information. But when you get it, that's it. I don't know what you want to call it. That's really the last word in it. Now things change in science. I mean, we started off with Newtonian mechanics, thinking that was the be-all and end-all, and of course quantum mechanics came in, and we realized that we didn't really quite understand everything about how systems work, and we've now amended it. And so science is always the latest. It's not really the last word. It's an ongoing effort, and we just have to keep probing to find out more and more. But what we find out is really, that's it. We have found it, and we can rely on that as the latest, best information we have. So when people talk about proof, it's a funny kind of issue. Scientifically, you don't really talk about proof. You talk about the best information that we have at a particular time, and that information has been verified experimentally. Cells contain a lot of different components, and not every cell has everything in it. And as you know, there are long cells like the nerves, which have long axons that make connections between different parts of the body. But the parts, they all have some kind, I shouldn't really say all, I keep thinking of exceptions. I say they all have a nucleus. We know that one of the commonest cells we all come across, the red blood cell, has no nucleus in its mature form. It's ejected the nucleus. But most cells, virtually all cells, have a nucleus. And it's the nucleus that has the DNA. And the DNA is the material that we shall focus on. The DNA, as we all know, is a long chemical. It's about 6 feet long inside us, and it's coiled up in 2 strands. The sugar phosphate strand, little sugar units connected by phosphates to other sugar units, and the 2 strands are joined by chemicals called bases. And the bases are links that hold the 2 pieces together, and these 2 pieces are twisted so that they end up making a helix. And we all know the double helix from the Watson book and the Watson and Crick work that was based on the experimental studies of Rosalind Franklin, who was not awarded part of the Nobel Prize because she had, unfortunately, she had died probably of results of exposure to radiation, which was part of her work, her research work. But anyway, the double helix is something that we know is the basic structure of this molecule, and the links between the 2 strands actually are part of a code. The bases, as they're called, tell the body which amino acids to put together when they make a protein. And so if you play games with the DNA and you change any of the bases, or you take a link out, or you break the chain, you are affecting the code. And that's why it's very important that the code remain intact. The second point I have there is that it's something we know that is passed on from generation to generation in order to transmit the characteristics, the human characteristics. So that's why children look like parents in most cases. But people seldom focus on the day-to-day kinds of things that DNA does. It is involved in protein synthesis. Protein synthesis is what keeps our bodies functioning. In other words, cells have to keep making proteins to replace those that are damaged, those that have to be made. In other words, when you eat something and you have to get enzymes into the gut to digest the food stuff, you've got to make more material. You've got to make more pepsin and trypsin and these kinds of things so that they will digest the stuff. And the way you make it is the DNA will come apart and start making these particular proteins. And if you have any damage that's done to the DNA, this has to be repaired as well, and more proteins are made in that process as well. Now, DNA damage can lead to cancer after many years, and that's the part that we are always focused on. And we will see that that's the part that's really very important for an individual. And when you get too much damage, it can lead to something permanent, the permanent damage being called a mutation. Now, mutations are bad for individuals, but they're not fatal in most cases because the damage is something that is lost when that particular cell dies. Cells are replaced, they become senescent, and the body gets rid of them. And you get new cells when the good cells divide. But damage is devastating frequently to the species, because if you get damage in a germ cell, that damage is transmitted to the next generation, and the generation is born with all kinds of bad things. And that's the part that we have to guard against. And people who worry about the damage to germ cells are really telling you that it's damage not to the individual so much as the damage to the species. Now, this is a picture of the DNA, and you see it's not the DNA molecule as a chemist would look at it, which is basically a long string. You see it's 2 meters long. It has 3 billion of these base pairs, and these base pairs are the parts that contain the information, the code. But the thing about this, the DNA in the nucleus, is that it has a particular structure, and it is coiled. We started off with the helix, and these little lines between the 2 parts of the helix are the bases that have this code in it. And it starts off as a twist. But then to pack it into this very small area in the nucleus, you have to keep coiling it. So you get this helix is then coiled. And then the coil itself becomes like a fiber, and that gets coiled. And you have coiled coils, and you keep on having this thing so that it's finally packed together in a very, very small volume. The interesting thing about the coiled coil is that this allows the structure to respond to many different lengths. I'm going to talk about the DNA as a kind of antenna. Antennas that tune in to different signals that are transmitted in the atmosphere are designed of particular length. If you remember when TV came in, some of you may remember that, they used to have 2 different antennas, 2 different metal bars. They had a long one and then they had a smaller one on top. They were for the 2 different frequency ranges, because each frequency has an optimal absorption. I mean, each length has an optimal absorption for a particular frequency. Well, it turns out that the coiling allows different lengths of the DNA to tune in to different frequencies that are being transmitted, or at least that's the way it appears to me. Because the DNA is not an inert molecule. Those little lines that I showed you at the bottom of this slide are really chemicals that have electrons distributed at their faces. So that when you have the 2 strings together and these links between the 2 strings, you have little like sheaths of electrons in there. These are electrons that are delocalized. In other words, they're not really locked into a chemical bond. They form like a cloud, an electron cloud. In fact, they're referred to as pi electrons. The electrons that will make a bond, let's say, in a hydrogen molecule between the 2 nuclei, those are special electrons that are locked in between the 2 nuclei. But the thing is, these pi electrons form what has been referred to in the literature as a pi wave. In other words, they can actually be a conduit and people have shown that these electrons can conduct. So you see this DNA structure has something in it that has the properties of a viable antenna. They can react to the signals that are being, that they're sensitive to, and they can react, because of the coil structure, they can react to a whole variety of different frequencies. Now, one of the things that has been reported, and it's been reported by many labs, is that EMF can break up DNA. This is a report now in the very weakest range. This is the ELF range, power frequency range, reported by Lay and Singh back in 1997. They showed that if you take a nucleus and you try and move it in an electric field, you can see that there are little bits and pieces that come off it after exposure to EMF. So if you look in the first the first screen there, you see that at that particular, in the control, you get little bits and pieces that are off, and normally from the handling that they will break off. But then if you go to the right of that, you see that more come off when it's exposed to a 1 Gauss exposure. This, by the way, is the level that is considered standard. You have to protect to this level where there are protections in place. 1 Gauss is the normal safety standard. So you see, even at that level, you get this kind of damage. And the same is true as you increase the strength of the field, you get more and more damage. The other thing that one finds with DNA is that there's a reaction. This is work that we did at Columbia. I say we, my colleague, Reba Goodman, and I have been working together for many years. And this is one of the things that we found. This is a review article in a specialized journal called Cell Stress and Chaperones, where we reviewed the earliest work on this problem. And we showed that when cells are exposed to very weak signals, comparable, actually much less than in the previous slide, Henry Lai and Narendra Singh showed with the breaking up of pieces from the nucleus, very, very low values. This is in the milligauss range, actually a few milligauss. You use something like 5 to 8 milligauss, you can get these, the activation of the stress response. The stress response is described as a situation where the DNA is made to come apart so that you can read the code, and the code is read in the body, and the particular proteins are made. These are stress proteins. There are about 20 different ones in nature, and the fact is that we followed one and found the particular DNA configuration at the level of the promoter that is involved in reacting to the EMF that is in the environment. This stress response occurs with a variety of harmful stimuli. It was first identified in response to heat. Cells cannot tolerate excessive heat. We, are homeotherms that have to be at a constant temperature, and the cells function at best at that level. And this is true of many cells. And if you raise the temperature, you find that these stress proteins are made. So the first identification of this stuff was as something called heat shock. That was the way this process was called. But we find that cells react not only to heat, they react to changes in acidity. If you go away from neutrality, they react to toxic metals. Cadmium is one of the things that's used to induce it. They react to alcohol. You may be interested to find out. And those of you who caught the program on the PBS, they're running a thing about prohibition, where alcohol was considered a moral kind of hazard. It's actually a biological hazard. If you get too much, you will get these stress proteins synthesized, indicating that your cells don't like that. It interferes with a lot of action, normal actions in the body. The stress response, and this is the last point I have on this slide, is that it differs from the normal way in which we use the word stress. When people talk about the body being stressed, we think about things like people getting scared, they start running places, Fear, for example, is something that will make you, but that's not the kind of stress we're talking about. That's the stress that will lead to the release of adrenaline, cortisol. These are the hormones that will get you to fight or flee. Those are the kinds of reactions that one gets to systemic stress. I'm talking about cellular stress. It's the cellular stress response that tells you that the cells are being harmed. And that's what we get with exposure to EMF. Now, I've talked about these two reactions and these that cause changes in the DNA, activation of the DNA. And one of the things is that they can actually damage the DNA. They can cause mutations that are similar to the ones caused by chemicals and radiation. But the fact is that when the DNA opens up and comes together again, it's made proteins in the process. And all these acts that are going on at the molecular level don't always function 100%. We're not perfect. And so you get errors that creep in. Now, in protein synthesis, the error rate may be 1 in a billion. But it may be, if it's a critical protein, that billion may be very important. And it may lead to some kind of malfunction. So that mutations can occur also in repair genes. And I have a reference down in the slide of a study that showed that these repair genes, mutations in repair genes, are correlated with the incidence of leukemia in children. Children who have imperfect repair genes cannot repair the damage that normal children can, and they have a higher risk of leukemia. So that was a very important paper that came out of China a couple of years ago. Now, the repair mechanisms are not always 100% efficient, as you might expect, and you get what are known as DNA deletions, that is, whole sections of the DNA can come out, and sometimes you get sections coming back in, or coming back in a different place, and all of these things lead to damage that can show up in later life as cancer. We know that this is true from experiments that have been done with twins. Now, twins, as you know, are identical copies. I'm talking about identical twins. They have the same DNA. The egg splits, the fertilized egg splits, and you get two individuals that have the same genome. The fact is that as you measure, if you measure the DNA after a number of years, you find they're not identical, because they're just the ordinary business of living will cause, there'll be some changes, and they have found that. And the other thing there have been studies on is autism, comparing autism in children and parents, and they find in those cases there are enough differences to let you know that these DNA repair is going on all the time, and that it's not always 100% efficient. Having given you this background of what's going on with the DNA all the time, and the fact that it has all these properties of being able to react in different lengths, we just published a paper last year saying that DNA acts as an antenna. Now, we're accustomed to antennas being long pieces of wire, but an antenna is something that can conduct electricity, or that is respond to electric or electromagnetic signals in the environment, but can also has these two properties, being a conductor and responding to different frequencies in the environment. And it's the different coil sizes that allow these differences, the response to a lot of these different differences. And the last point I have on this slide is that this greater reactivity range means that DNA can be damaged by a wide range of frequencies. This is an important practical conclusion, because it means that whenever you set up a safety standard, the safety standard is always tested at a particular frequency. And you say, well, this will be safe at this level. But you're not exposed to that level only. You're always exposed to all the things that are around. And everybody's focused on smart meters or cell phones. But we've got the power frequency around us all the time. We've got TVs and RF frequencies coming out from transmission all the time. I've talked to high school classes. I go in with my little portable Sony that has 15 bands on it that can pick up shortwave and radio frequency and FM and all that. and I can tune in, get a signal on each one of them. The power comes from the batteries that I put in there, but it picks up the signal that's coming out through the atmosphere, and I'm tuning into it with this little device. Well, our bodies are picking up the signals that are around as well, and we have the energy to do things in response to those signals, because we have our own batteries inside us that are giving us the energy to do that. So I have here a summary, kind of a checklist of the kinds of experiments that have been done that indicate the ranges in which these two particular effects that I concentrated on, the heat shock that's in the upper part, the red, the reaction with DNA, HSP are heat shock proteins, the changes that occur with heat shock, and also the DNA breaks that occur. And you see there are check marks next to all of the major groupings on the EM spectrum. And the one in the middle, on cell phones and Wi-Fi, you see is also checked. So people have shown that these things occur across the EM spectrum. And this, I think, to me, is an indication of the property that are referred to as a fractal antenna. When this occurs, when engineers make antennas that can cover a wide range, this is indicative of a special property, and our DNA apparently meets these criteria. Now, we published this paper this past year, and not everybody's happy with it. And there was a rebuttal that came out, and I thought it was a terrible one, and I told the editor not to publish it. And so the man cleaned up the paper, and he responded to some of the points that I made, and he sent in another letter, and I read it, and it wasn't, I didn't think it was worth publishing, but the editor thought it would be a good idea. So he asked me, he said, would you mind if I published it and with your rebuttal? So I said, well, sometimes controversy is useful in science. It might highlight an issue, and so it will appear in the International Journal of Radiation Biology. So you will see I don't know, it will be published there. The rebuttal is, you'll see from my rejoinder, my response to the rebuttal that I still think it's without merit, but the fact is it will appear. So far, I've concentrated on the DNA stuff, and I think, as you can see, there's a lot of information there. To me, the information is very strong. I haven't gone into other stuff which we've done. We found effects on enzyme rates, very important enzymes. The sodium potassium ATPase, which is the basic enzyme that pumps ions across the cell membrane and is responsible for building up a membrane potential that allows cells to function, allow nerves and muscles to function. Also, we showed the magnetic fields can affect the cytochrome oxidase, which is a vital enzyme in mitochondria that allows these cells to build up ATP so they can do their work. That's the basic fuel in biochemical reactions. We've published that. That's in the literature, but I haven't focused on that. What I've done is mainly show you about the DNA. But there have been many other studies that have been done that show that there's damage to nerves, damage in sperm cells. There's radio frequency broadcasting increases the risk of cancer. Cell phones increase the risk of brain tumors and salivary gland tumors. And there are increases in leukemia, Alzheimer's, and neurological diseases. The last point having to do primarily with the lower frequency range. Here's the RF broadcast. This is a study that was published online a while back by Neil Cherry, which shows the incidence of cancer in this region in San Francisco. And this is over a long period of time, from 1937 to 1988. These are data that are in the literature. And these are, in many ways, it's the kind of study that was done, that was published by Sam Miller on dirty electricity. He just got the information that was in the records. Nobody went out and set up a study and got case of controls and all that. They just saw what the death rate was in the region of children who live in the region of Sutro Tower. And they found that, as you can see, there's a fall off of the incidence, the risk ratio, RR is risk ratio, of developing cancer as you go further and further away from the antenna. Now, if you look at the point at 3 kilometers from the antenna, it's about 2 miles away, you get, they measured what the actual intensity was. And the intensity at that point was 1 microwatt per square centimeter. Now, this is 1,000 times lower than the rate that is considered safe. In other words, the safety standard is set at 1,000 microwatts per square centimeter. And these data were for a distance of 3 kilometers is at 1 microwatt per square centimeter. And the risk ratio was still 5. That's a pretty high risk ratio in epidemiology studies. Now, this is, I think, a very strong indication that just living in the vicinity of a RF broadcasting system constitutes an added risk. And it should give everybody pause when they talk about what's going on with cell phones, because you're getting this all the time you're in that vicinity, whether you use a cell phone or not. Now, there's a recent study that came out with just this year in Brazil, in Belo Horizonte, and they did a study of all cancers in that particular town or city, and they found that it was those living within 500 meters of antennas have a much higher mortality rate. They compared the 500 within 500 meters to those outside that radius. The safety limits that they have in Brazil are set at 2 different frequencies. But in Porto Alegre, which is in the same vicinity, the same state in Brazil, they have actually lowered the level to 4.2 microwatts per centimeter squared. They have recognized that there is a problem associated with this kind of tower, exposure to tower radiation, and they tried to lower it. But as you can see from the list below, it's not as low as some of the other places that have set limits on the kind of intensities that are being allowed. The lowest, of course, is set in Salzburg, in Austria. And that Salzburg set the level at a point that was recommended by the Bioinitiative report, which I'll come to briefly in a little while. This is another paper that came out this year. This is a study from Israel where they initially showed the effects on salivary gland tumors. Now, salivary gland is located just where you put your phone. It secretes saliva into the oral cavity when you're eating, and there are a whole bunch of different salivary glands. And this study showed that when you measure they collected data for the incidence of salivary gland tumors when they compared the tumors in 2 different areas. One area is the normal parotid gland, which is located above the mandible, and the other is the submandibular gland, which is located below the mandible being the jawbone. And the thing is that if you are below the jawbone, the gland is protected by the bone density, so that it doesn't get quite the radiation that the other one gets. And you can see the submandibular stays pretty constant with the introduction of cell phones, but the other one goes up, and keeps on going up, and leads most people to be very pessimistic about what's apt to happen with the passage of time and the increased use of devices. Now, just a brief word about power frequency in disease. I haven't focused on this. This used to be old stuff, but the fact is, people have known for a long time that there's an increase in the risk with leukemia, with exposure, and the exposure is at very low levels, 3 to 4 milligauss. That's very, very low. The repair gene I already mentioned. Study in Switzerland has shown that Alzheimer increases for people who live within 50 meters of a power line. this is a huge study that was, I say huge, because it encompasses a large percentage of the population of Switzerland. And the fact is that they have this enormous increase that shows not only an increase, but has the characteristic that epidemiologists like to find, which is a dose response. So if you have a 5-year measure of the effects of 5 years exposure compared to 10 years, it goes up. And then at 15 years, it goes up still further. And at 15 years, living within 50 meters of the power lines, you double the rate of Alzheimer's in the population. So that's a pretty clear indication epidemiologically that there is a great risk. Breast cancer is another point that is huge research on that, but huge controversy as well. I mentioned a paper by Roberti who found at the cellular level that there is an indication that EMF at the low frequency can affect the activity of breast cancer cells. And it can actually not only affect the activity, but it can, if you use an inhibitor, which is tamoxifen, that's been used in cases of breast cancer to decrease the recurrence of breast cancer. When you expose these breast cells with tamoxifen to an electromagnetic field, you overcome the inhibition that's due to tamoxifen, and the breast cancer cells grow. So that it's a double hazard. That is, it may induce breast cancer, but it can also affect the therapy that's being used to inhibit the growth of the breast cancer. And the last point I have here is the point about the Millen book, which is what I mentioned earlier, where he has shown that with the introduction of electricity in the U.S. in the 20th century, he collected data from different parts of the country and compared urban regions to rural regions where the electrification occurred in the different areas, compared the north to the south, and it showed that with electrification there was an increase in cardiovascular disease, cancer, diabetes, and suicide. You may say that these are numbers, but these are numbers that were not manipulated. They were collected by people who were just writing down, like Jack Webb used to say on the television, just the facts, ma'am. These are just the facts. They put them down without any hypothesis and without any agenda. And he just looked at them and said that this is the way it turns out. And this kind of study, by the way, is more in keeping with what epidemiology started out to be. If one takes a look at the very first epidemiology study, there's a great book that's out called The Ghost Map. I don't know how many of you have read it. It deals with the epidemiology study, well, with the study that was done by Dr. Snow in London, the cholera outbreak in 1865. And he showed where the cases were, where the cholera cases were showing up in the Soho region of London. And he made a map of it, which is where the book gets its title. And the thing about the map is that it focused on right around the Broad Street pump, the water pump, which is where people were getting their water from. And he just said, well, gotta do something about it. And he did. He just removed the handle so nobody could use it. He didn't get any permission from the authorities. And by the way, the authorities believed that it wasn't, it took, I think it was about 20 years later that people finally realized the germ theory was effective, it meant something. And at the time, everybody thought that cholera was due to a miasma, foul air in the atmosphere. So he wasn't operating on any theory, but he did the first epidemiology study, and he was convinced enough to actually take action. And he did the action, and the cholera abated. But nobody gave him credit at the time, and I think he wasn't given any credit. I think he may have died before the germ theory was set. But anyway, this is the kind of study that Sam Millen has done in the spirit of what epidemiology started out to be, and he's done it in a way that makes it, that really gets interesting material that is both important and that can be acted on. But let me just say that EMF, from a scientific point of view, is a health risk. And you see the exclamation point on there. There are biological mechanisms People say there are no mechanisms. There are many mechanisms. The very first mechanism, by the way, was pointed out by one of the earliest studies that were done in the 1970s, where they showed the leakage of the blood-brain barrier. This is work by Alan Fry that was overlooked, in effect. Showed that just exposing the head caused the leakage and recently was shown that this results in death of certain brain cells. There are epidemiology studies, some of which I've shown you. And the important thing, I think, is the third point here, that the safety standards do not protect. They're based on the energy and the energy of a EMF wave, and the fact is that the energy is unimportant. Apparently it goes to EMF of all energies. That the energies increase as you increase the frequency in the spectrum, and we find that these kinds of biological effects are across the spectrum. We must limit exposure to EMF if we are to protect the population. And the last point I say there is that precautionary measures are needed. Precautionary measures are those that are taken when people are not entirely convinced, but they're cost-effective measures, like things that can be done at relatively little cost, because the price you pay afterwards, the damage that's done by not taking these measures, cannot be measured in dollars. Thank you.